Good morning, everyone. My name is Niek, and I'm the European ambassador to the International Genetically Engineered Machine Competition, or iGEM for short. I'm going to be talking about safeguarding the future of biotechnology, the lessons that we learn in iGEM. I'm pretty sure that some of you know what iGEM is. For those of you who don't, it will become apparent during the talk. But first, I'm going to show you this. This is a brilliant picture, a famous picture from the Caltech archives of the blackboard of Dr. Richard Feynman. As you know, he was one of the people in the Manhattan Project and won a Nobel Prize in physics. And in the top left corner, you see his most famous quote, "What I cannot create, I do not understand." I feel like Robert Langdon doing this. It's amazing. So this quote, it's often taken, and the literary meaning of it is reversed to evoke some kind of an argument. See, this polarizing view can reflect on things like genetic engineering, or biotechnology, or synthetic biology. See, these fields, they have something in common. It is often very hard to completely understand what we're doing. See, I don't completely understand what I'm doing in the lab. I don't have to explain it to you, for instance. We can see stuff happening where we don't completely understand it. But what we do understand is that these technologies will shape and define our future in health and in medicine. So in order to strive for a bright future, we need to work on understanding what we're doing. And the best way to do that is to teach students a proper way of safely handling these powerful technologies. Because students will not just become the next scientists, they will also become the next communicators that will bridge the gaps between science and society. And this is something that comes back in this amazing competition. So it's a competition for anyone who's interested in synthetic biology. And to give you a short little recap, each year over 5,000 students from 350 universities, colleges, community labs, and most importantly also high schools is on the rise. They work for over 8 months, 4 to 8 months it says here, on a project in which they solve a local problem using synthetic biology. Now, what does that mean? I'm going to analyze that for you in 4 distinct levels. First off, synthetic biology. At its core, this competition is a competition in genetic engineering. And these students, they spend 8 months in the lab working very hard. And this synthetic biology, it's very closely guided by safety and security. We want these students to do this safely, to not harm themselves, and also not harm the environment. Next up is human practices. And this asks the question, should I be doing this? This is a question that is very often not asked when people are working in a thesis. So it's completely unique here. And lastly, we want these students to become communicators. They have to go outside and have to talk about their project. They have to become the experts that can explain anything about their project. So let's zoom in a bit and let's give some examples. So finding a solution to a local problem. This one, brilliant example. It's a team from Wageningen University, that's the agricultural university in the Netherlands, and they worked on a problem. See, everyone loves bananas, and they found out that there was a disease that was tackling these bananas, called Panama disease. And they engineered a bacteria in such a way that it could detect this fungus and would produce a toxin for it. So that's a brilliant example of solving a local problem. And this solving a local problem, the competition, the synthetic biology part, is largely based on building standardized parts, building a community. So just like the seed vault that we have, we're creating a very large repository that has been, I think, opened in 2003 and built from ever since, of biological parts that anyone in the iGEM competition and anyone in academic research can use and build on. Next up, the interesting part for you, safety and security. So I don't have to explain here that regulations within countries are completely different everywhere. So it's very hard when you have 350 teams from 50 different countries to get them into one single guideline. So we start off very easily. They have to follow their university guidelines. Next up, the regional and national guidelines that are set by the countries that they work in, and the worldwide regulations that are usually made in this city. And lastly, the iGEM rules. See, we have our own set of rules. And we constantly adapt these rules because these students, they're often working with technologies that haven't been published yet. They're working with gene drives 2 months after the first paper was published on gene drive. That's amazing to see the ingenuity that these students bring to this competition. And we have a very strict rule that I should start off with. The rule number 1 is do not release. Well, this is a competition that is focused on showing the potential of what you're doing. We don't want these students to bring genetically modified organisms into the wild. So again, an example of what is safety in iGEM, because we do want them to think about it. So this is a brilliant example of safe by design, of responsible innovation. This was a team that made a device that you could hang inside of a beehive, and it would kill the Varroa destructor mite that was killing the bees, and it's still doing that all over the world, causing major losses to bee population. But they found out that if you have a bee that lands on this device, which has a genetically modified organism on it, bees will spread it everywhere. So they made a kill switch that is regulated by sunlight. As soon as the bee would leave the beehive, the sunlight would kill the bacteria. And it's ingenious, and this kill switch has been added to this repository, so it's free to use for people. Another great example that actually builds on your talk, the talk before me, is the dual use. So this team, they discovered something pretty brilliant. You can order synthetic DNA from large synthesis companies. And we don't want anyone in the world to be able to order the entire genome of Ebola or smallpox. So we put in very strict guidelines and methods to check what's happening and what is being ordered and if there are licenses for it. And this team, they did some very smart engineering. They found out that you can encrypt DNA in such a way that these synthesis companies wouldn't pick it up if you ordered sequences that were illegal. And what they did is they reached out to us at iGEM. They reached out to the companies, and they worked with them to fix this problem so it couldn't happen in the future. So that's a brilliant example of what iGEM stands for. See, we don't just punish bad behavior. Don't get me wrong. If there are teams who are not following the rules, we kick them out. But we also reward good behavior. We put these teams on a pedestal because this is brilliant that I found this and no one else would have found it. And it's very good for the competition. So this is all being there's a committee of 35 individuals who watch these iGEM teams and guide them and work with them from all kinds of institutes over the world, and there might be some in this room actually, I don't know for sure, who work with them to make sure that what they're doing is safe. So let's talk about the next layer, this is human practices, and this is asking the teams, should you be doing what you're doing? And while there are many hundreds of examples I can give you, I'm going to focus a little bit more towards the policy side of this. This team worked with, it's from China, worked with a few Chinese industries, as well as European governments, to see what the differences are in biosafety laws. And they wrote a very interesting paper on it and published it, and we gave them a prize for it. Or another one, there was a team from Heidelberg who found out that what they were working with in the lab could produce harmful sequences, laboratory evolution. And they made an artificial intelligence tool that could predict these hazards. And they made it open source and available for anyone to use. Or education. Here we see little kids playing with, or not playing with, being educated about the building blocks of life. Because that's where it all starts. That's where enthusiasm starts. So lastly, communication. These teams, they work an entire 8 months, and they go to the Jamboree. That's the giant event in Boston. It's actually 1 month from today. So most teams are pretty stressed out right now. And we don't want them to stay in the lab these entire 8 months. No, we want them to go outside. They organize debates. This is a team from Delft who did a train debate. They give TED Talks. But they need to, because they need to practice it, because they go to Boston in November. And here, they have to convince the jury what they're doing. They have to convince them in a poster presentation. They have to present in front of hundreds of people. And if they're lucky, they belong to the 6 finalists who get up on stage in front of these 5,000 or 6,000 people. And that here, they can show them that they understood what they created. Now, it doesn't just end here. Oh, wait. There's supposed to be a large picture here, but that's gone. OK, so it doesn't end there. This competition started in 2006 at MIT and has since grown to 50,000 alumni and 5,000 projects. This competition creates highly motivated individuals who want to build this world of synthetic biology, who want to work together, who's been through the same process, who's created the same mindset of thinking about things in their project to a very different degree than they would have done if they would have just followed their normal study program. So I urge you that if you ever get into contact with one of these teams, listen to them, embrace their enthusiasm, because it is those people who will at some point end up in this room as being the major decision makers in the world of biotechnology. Thank you very much.